want to welcome everyone here to a special event. My name is Josh LeBert. I am the Executive Director at the Biodesign Institute, and tonight is the Arndt Grand Challenges Lecture. It's an annual event that we have here. Ordinarily, we would be inviting you all to come visit us at the Biodesign Institute, where you could actually be part of our whole program on the university campus. I suppose the one benefit of tonight is that we'll be able to get more people to the lecture because we have a limited size lecture hall at the university. But we certainly wish we could all have you in person and there would be food and other things. So welcome to a different event. I want to mention, first of all, that after the lecture tonight, there will be questions and answers. And if you'd like to have any questions answered, please add them to the chat down below at the bottom of your Zoom screen. We'll be taking all of our questions from there. So please add them to the Q&A at the thread down at the bottom. Just for those of you who don't know Biodesign real well, we are an institute within Arizona State University. We are specifically what we call a translational institute. Our goal is to do research that has impact and helps solve the world's problems. And so we are organized into centers, and each center has a focus on solving some kind of world problem. We are very multidisciplinary in nature. That means that we have people in our centers from all over the university, from medical doctors to software designers, from engineers to molecular biologists, physicians, all sorts of researchers of all kinds working together to solve big problems. Some of you know that our institute has been very busy working in the context of the COVID crisis. And as you saw briefly in that sizzle reel, we have designed a saliva test that we are using to help test people in the state of Arizona to make sure that we stay safe from the virus. We're now typically doing somewhere around 4,000 to 5,000 tests a day. We are one of the largest, if not the largest, tester for COVID in the state of Arizona now, working in counties all over the state with free public testing, so help keep people safe. So tonight is a special night. It is the Arndt Grand Challenges lecture night. This is based on our founding director, Charlie Arndt, who retired despite our strongest objections in 2017 after a truly distinguished career, a National Academy member who did a lot of research based in plant molecular biology and used that plant molecular biology in novel ways. As you saw briefly in the sizzle reel, his work led to the development of a vaccine for Ebola. He is on the President's Council of Advisors. He under the George W. Bush administration. I mentioned that he is already a member of the National Academies of Science. Charlie, we're going to have you wave to us. I don't know if we can spotlight Charlie briefly so he can wave. He wants us to keep moving in the presentation, so we're not going to stop and chat with him for the moment. So it's my distinct pleasure to introduce to you Britt Glaunsinger, who is the featured speaker tonight. She's an endowed chair and professor in the Department of Plant and Molecular Biology, as well as the Molecular and Cellular Biology at one of the most distinguished universities in the world, Berkeley. I say that because I'm a graduate there, so I'm quite proud of that. She's also an investigator of the Howard Hughes Medical Institute. She also has Arizona ties. She did her undergraduate degree here at the University of Arizona in Arizona, and then her PhD work at the University of California in San Francisco, where I also did a degree. She is broadly interested in how viruses interface and hijack cellular pathways that control gene expression. Many of you know viruses are these little things. They're not really quite life. But they co-evolved with humankind from the very beginning and have developed very clever ways of getting into our cells and taking over things. She's an expert in her field. She's given a number of lectures on this topic, and we're very happy to welcome her tonight to teach us more about what's going on with the coronavirus. So with that, I'm going to hand it over to her.
Thank you, Josh. I'm going to go ahead and share my screen now. All right, and I just want to begin by thanking you for the invitation. It's a real honor to be able to present tonight for this exciting Arndt Grand Challenges lecture series. As Josh mentioned, for pretty much my entire career, I've researched how viruses interact with and change the environment of a host cell during infection. And for most of my career, I've focused on DNA viruses in this context. But over the last year, like many research labs, we've focused also on coronaviruses and their biology. But for tonight's lecture, I don't want to go in and focus specifically on the research that my lab is doing. Instead, I want to go broader to distill for you some of the leading edge research that's being carried out all over the world to understand the biology of this virus and figure out how we can fight it. And I'm going to break my talk tonight into 3 sections that are about 10 minutes each. I'm going to begin with a discussion of how SARS coronavirus 2, or CoV-2 as I'll refer to it, enters your cells and is able to replicate inside of your cells. Then I'll discuss what influences, in particular, what aspects of the immune system influence COVID-19 disease severity. And then I'll close out by going over the landscape of therapeutics and vaccines that we have to fight the pandemic now. But let me first start with an overview of the virus and the viral particle itself. What comprises the CoV-2 virion? Well, like all viruses, this virus has a set of genetic instructions that basically have all of the information needed to tell the cell how to convert itself essentially into a virus-making factory to pump out hundreds or thousands of coronavirus particles. In this image, that genome is a set of instructions on a strand of RNA shown in this orange spaghetti-like structure here in the diagram. For coronaviruses, that RNA genome is remarkably long. In fact, it's much longer than almost all other known RNA viruses. And in a few minutes, I'm going to tell you why that probably is the case. This RNA genome in the viral particle is protected by these blue structures. These represent another viral protein called the nucleoprotein. And this RNA genome is surrounded by a coat that's a lipid envelope shown here in tan. This protects the genome from the elements and is stolen from the host. Many viruses use lipids or membranes in their biology, but no virus has the capacity to make or synthesize its own lipids. And so they all have to essentially hijack or steal them from the host cell, as the coronavirus does in forming these viral particles. Studded within that viral envelope are a number of different viral proteins, some of which I won't talk much about, like the matrix protein, which is shown here in purple, or a minor envelope protein, which is shown in yellow. However, I will spend some time talking about the most famous of the coronavirus proteins, this very prominent spike structure in orange, which is what gives the particle its name. So when scientists were originally looking at these viruses under an electron microscope, what they saw was an array of structures that looked crown-like because of these spikes emanating from that viral particle. And those spikes, or coronas, are why this virus is called the coronavirus. The spike protein is the focus of nearly every one of the vaccine efforts that are ongoing right now. And that is because this protein plays 2 absolutely essential roles in the viral biology. That of identifying and docking onto cells, usually in your lung, that it can get into. But second, it also plays a key role in allowing the virus to slip past that outer barrier of the cell, the plasma membrane, to deposit its genome payload into the cytoplasm of the cell. Let's go over how it does this. So you can think of the spike protein as being like a viral key, and that key needs to identify a lock on the surface of the cell that it can fit into appropriately that will enable it to get inside. The locks on cells are called receptors, and the receptor for CoV-2 is this protein called ACE2 on the surface of the cell. Now, different viruses will have evolved to use different receptors as their lock to fit one of the viral protein keys. These are proteins that are, of course, not there on your host cell as a means to let viruses in, they all have other functions in our bodies. ACE2, for example, has roles in regulating viral blood pressure, host blood pressure. Nonetheless, coronavirus, through reasons that we don't really understand, has evolved to use this receptor. And so the spike, or the viral key essentially, will dock onto this receptor as a means of identifying that this is a cell it has the capacity of getting into, because not all cells have this lock on them. But you'll notice a second thing happened when it docked onto that ACE2 receptor, was that it looked like this tip of the spike protein was cleaved off. And indeed, that does happen. There is a protein called a protease enzyme. This is like a pair of molecular scissors. It's also encoded by your own cell. This set of molecular scissors, the protease enzyme, will come and cleave off the tip, the head of that spike protein. The reason it does this is that there's a second crucial activity housed in this spike complex. And that is a spring-loaded like fusion machine that is tucked not in the tip of the spike protein, but more in this lower stalk region of the protein, closer to the viral core. You can think of this as like almost a jack-in-the-box. The spring is in a condensed state, inactive, and held in that state because the lid is on. And that lid is that surface tip of the spike protein. And so in order to turn on this fusion machine, the lid has to be opened. And when that happens, now this, what's called a fusion peptide, unfolds from the spike protein, inserts into the plasma membrane of the cell, brings the cellular membrane in close proximity to the viral membrane so that the two can mix, forming a portal through which the viral RNA genome can now enter the host cytoplasm. This is the start of the infection. When in the cytoplasm, that host RNA has all of the instructions needed to make many, many more copies of the coronavirus. And it is read by our own protein synthesis machinery, a complex called the ribosome. That ribosome makes all of the coronavirus proteins that the instructions tell it to, and those proteins do the work of converting the cell essentially into a virus-making factory. So as you can envision, there's a lot of changes that are happening to the inside of that host cell during the viral infection. This on the left is an electron micrograph image from Montserrat Barcena's lab, where she has infected cells with a coronavirus closely related to CoV-2. And what you'll notice is that in the inside of this cell, there are a whole bunch of these ring-like structures that look like a ring within a ring. And indeed, these are membranes, double membrane structures. When taking these microscope images, they can take a number of stacks of images, allowing the scientists to reconstruct in 3 dimensions what the interior of the cell looks like. And that 3D reconstruction is shown here in the cartoon on the right. And so these are in fact large spheres that are being formed by the virus during infection. These spheres are made of membranes, host membranes, that viral proteins have stolen and reshaped. So essentially, some of the viral proteins will go to the intracellular organelles, like the endoplasmic reticulum, take that membrane, pull it out, stretch it around, and use it to form these new spheres in the cytoplasm. These are like separate rooms in the cell where the virus can then amplify its RNA in a protected region of the cell, perhaps out of the view of cellular factors that are trying to stop viral infection. Now, one of the exciting things that was discovered from this study is that within these spheres, the virus actually forms these special portals here, shown in the red, that allow the RNA that's being made to transit out of the spheres so that it can be translated by the host ribosome, the protein synthesis machine. These are these purple structures here. And also perhaps transit to new vesicular structures where it can be enveloped to form novel virions that will then be released from the cell to go on infect neighboring cells, or to be breathed out by you if you're infected so that they can infect a new host. I want to spend a minute also talking about what's inside of these spheres. In green here, this represents the viral RNA genome, which is amplifying like crazy in these rooms. And this amplification of the RNA genome is being carried out by a special enzyme that is an RNA copying enzyme called a polymerase, an RNA-dependent RNA polymerase. All RNA viruses have to bring in or encode their own polymerase enzyme because unlike things like the protein synthesis machinery or the lipids, our cells don't have any enzyme like this that they can use and steal to copy their RNA. So they've got to bring their own. The coronavirus RNA copying machine is remarkably sophisticated. In this cartoon here on the right, you can see that it's got a core copier, which is this RNA-dependent RNA polymerase designated as NSP12, that's the name of the viral protein. But it's got at least 9 other viral proteins that come together as a complex to make this copying machine. These proteins are helping the copier maybe move faster, be more accurate, to make additions or modifications to the RNA as it's being copied so that that RNA can be more easily recognized by the protein synthesis machine to pump out more coronavirus proteins. There's a second very unique feature about the coronavirus RNA copying machine. To understand why that's unique, first I have to tell you that all RNA copying machines make lots of mistakes. This is a hallmark feature of RNA viruses. And this random errors that as they're going along copying the RNA, they accidentally will insert the wrong nucleotide periodically. This is the basis for the rapid evolution that happens of RNA viruses that acquire lots of mutations that allow them to escape things like antibodies and therapies that you hear about for viruses like influenza and HIV. It turns out that unlike almost every other known RNA virus, the coronavirus RNA copying machine has the ability to correct its errors. It has a proofreading capacity. So as the enzyme is going along copying the RNA, it's going to make errors just like every other RNA polymerase does. But unlike every other RNA virus, these viruses can have a mechanism to recognize those errors, kick out that incorrect piece when it's inserted, and put in the correct piece. So instead of having a genome that is studded with lots of mistakes, the coronavirus genome is going to be much closer to mistake-free. So from a human perspective, this might benefit us because you could think that fewer random mutations that are made in a virus might mean that fewer mutations could arise that are going to allow this virus to escape from neutralizing antibodies or to make a vaccine less efficacious over time if the virus changes very rapidly, like what happens with flu, and that's why you have to get a new flu vaccine every year. From the viral perspective, though, what it means for the virus is that it enables the coronavirus to have a much larger genome than almost all other types of RNA viruses. And so you can sort of see this on the graph down below here, where the size of the coronavirus genome, nearly 30,000 base pairs, is 2 to 3 times the size of almost all other RNA viruses. And that's because the size of the virus is dictated often by the error rate. The longer you are, the more errors you might acquire, and that will make for a virus that doesn't work very well over time. So because it can correct these errors, it can have a much larger genome. That means that coronaviruses can encode more viral genes than other RNA viruses and have perhaps more sophisticated interactions with the host and the host cells. So let's talk about some of these virus-host interactions as they relate to immunity and to disease severity, and in particular to this highly researched question of why is it that some people experience asymptomatic infection when they get CoV-2, whereas others develop severe COVID-19 disease? Well, we know both from studies with COVID-19, but also from studies with other past coronavirus outbreaks, like the original SARS coronavirus epidemic, that severe disease probably arises when your body's very early initial immune response, which is called the innate immune response or innate antiviral response, is inefficient or insufficient. This then leads other components of your immunity to kick into overdrive in a bad way for you. This leads to overexuberant inflammation as your body sort of tries to compensate for that lack of a robust initial response. And this is what can drive severe disease. So let me show you what I mean by that. In the graph here on the left, I'm showing an example of what the viral levels and the immune levels might look like for an individual who has asymptomatic CoV-2 or a mild case of COVID-19. As soon as the body detects the virus at all, the virus is shown here in green, it is going to mount very early on what's called an innate immune response, and a robust innate immune response that is going to do a number of things to help kick an appropriate immune fighting machine into action. One of the things that it does is help kick down the levels of viral amplification and prevent viral spread. Another thing that it does is recruit in a regulated way an appropriate set of other cell types that are related to inflammation that will help clear those infected cells and get rid of the remnants of disease, and then of course later on establish long-term immunity. However, this is not the situation for all individuals. Individuals that have severe COVID-19, their initial reaction appears to be different. Instead of immediately mounting this early innate immune response, there's a delay before that innate immune response is mounted, and it tends to be mounted in sort of a dampened way. This is bad because not only could the virus in theory start amplifying to higher levels, but it's also bad because your immune system then tries to compensate for that by bringing in many inflammatory cells to try and fight the viral infection. But this is an example of too much, too late, because by the time all of these inflammatory cells make their way to the lung, well, the viral titer, the viral levels are already on the decline. And so these cells aren't so much needed to kill the virally infected cells. And instead, what they're causing is a lot of collateral damage, harming uninfected cells and uninfected tissue. And this is leading to hyperinflammation and what's called immunopathology, which is your immune system is causing the disease rather than the virus at this point. So because this very early immune response plays a critical role in the outcome of COVID-19, I want to spend a couple more minutes talking about what it is. So this innate early immune response is essentially driven by a series of what are called pattern recognition receptors. These are sensors that are present on the surface of your cells and on the inside of your cells that are constantly looking for foreign molecules. They're screening for things like bits of viral RNA or viral DNA that don't look the same as our own genome does, looking for bits of protein or lipids that might come from bacteria that aren't present in our human cells. And as soon as they detect one of these foreign molecules, these are going to send an immediate alarm system. And that alarm system operates through the release of these signaling molecules called cytokines and interferons. What interferons are doing for you as soon as they're released early on is at least threefold. They're going to send signals to neighboring cells that are not yet infected, telling them, hey, a virus is around, you might not be infected yet, but you better build up your antiviral arsenal to be ready when it comes. They're going to send a signal to cells that are already infected, telling them essentially, kill yourself now before the virus has a chance to finish its replication cycle and send out hundreds of new viral particles to attack other cells. These interferons are also going to help recruit white blood cells, which have components of the immune system that will help generate long-lasting immunity in B cells and T cells. So We know that these interferons are important, and one of the reasons as a virologist that I really like to use to explain how we know they're important is the amount basically of energy that a virus dedicates to trying to block this pathway from being activated. Turns out that CoV-2 launches a multi-pronged attack to dampen that interferon induction. These are just a few examples of many, actually, that probably the virus uses. This is a diagram that I've taken from a very recent report from the Goodman lab showing a few strategies that CoV-2 uses to block this. One of its proteins is able to inhibit the important sort of processing steps to produce the messenger RNAs that are needed that encode interferon-stimulated antiviral genes. These are hundreds of genes that get turned on in response to interferon signals that help cells set up an antiviral environment to prevent infection. There's another protein called NSP1 that is able to bind to the cellular protein-making machine, the ribosome, and selectively impair its ability to make cellular proteins that might be encoded by these interferon-activated antiviral genes, but still allow production of viral proteins. There are other proteins like NSP8 and NSP9 here that interface with the cell's protein secretion machinery and block the transport of vesicles that would be used to release interferon and cytokines out of the cell so that they can go and warn neighboring cells. So the virus is really trying to dampen this innate immune activation. But I started this discussion with, well, what's the difference between an asymptomatic infection and a severe COVID-19 infection? Because in theory, the virus is doing this in all of those cases. So why some people are then progressing to severe disease? Well, there are multiple studies that are now emerging looking at host genetics as to try and identify risk factors that may be present in our own genes to explain why some people are getting severe disease and others don't. I'm going to highlight a couple of recent examples that have come out of the National Institutes of Health and the Rockefeller Institute, in which scientists have looked by first sequencing efforts to look at the genome or exome sequences of humans that are either experiencing life-threatening COVID-19 or asymptomatic or mild COVID-19. And what they were looking for was evidence that there might be mutations or changes to the genes, the genome of these individuals that rendered them more susceptible. The researchers knew that interferon played a key role in this early response. And they also knew that there were interferon genes in that pathway that had previously been linked to susceptibility to other types of viral infections like influenza. So they specifically focused on this interferon pathway. And indeed, they found that in about 3.5% of patients that have life-threatening COVID-19, these patients have small mutations in genes that are important for interferon pathway function. They did not find any of these mutations in the patients that they looked at that had asymptomatic or mild infection. The second study, instead of looking at changes in the genes, looked at the chances that people might be producing a bad kind of antibody. These are called autoantibodies. Normally, as you know, antibodies are there to try and fight off infection of foreign pathogens. But sometimes some of our cells make mistakes and they put out antibodies that instead of recognizing foreign molecules, recognize our own molecules. This is essentially the body fighting itself, and this is the underlying basis for autoimmune disease. What they found was that in patients, about 10% of patients that are experiencing life-threatening COVID-19 in the cohort they looked at, which was nearly 1,000 patients, 10% of them produced autoantibodies against this type 1 interferon molecule. None of the patients that they looked at in the asymptomatic or mild category did. Interestingly, nearly all of these patients that produced these autoantibodies against their own interferon system were men. And we know that males have a predisposition for severe COVID-19, and this may partly explain that predisposition compared to females. And so to summarize what this means, that generally our body, the early responses are trying to use this type 1 interferon-based immunity to ramp up and get rid of the virus before too much damage is done. The virus itself is trying to fight this response so it does not get stamped out by blocking production of these antiviral interferon-stimulated genes. So most of the time, we in the population can overcome this viral antagonism and successfully fight infection nonetheless. But in certain cohorts of the population, individuals may have genetic mutations that impair their ability to use this pathway, mutations in components of this interferon signaling pathway, or their body inadvertently or inappropriately produces antibodies that instead of recognizing foreign molecules, actually recognize and inactivate their own interferon molecules, essentially dampening this later signaling that allows the body to mount an appropriate immune response. So, how can we fight back? Well, there are two classes in my mind of COVID-19 therapies. These are therapies that either target the virus itself and try and stop it, or therapies that target the patient's immune system, because as I told you, the immune system is the culprit later on during severe disease. So virus targeting therapies are going to be things like small molecules that will somehow bind and inhibit the enzymes of the virus or the proteins of the virus. For example, the viral RNA copying machine, the polymerase. Remdesivir is an example of one of these types of therapies. Other antiviral therapies are going to be things like monoclonal antibodies that are synthesized in the lab and combined. Usually you hear about monoclonal antibody cocktails that are designed to be specifically targeting the spike protein in a way that will neutralize it. So if these are delivered to a patient, they can bind the viral protein, the viral spike protein on a virion and prevent that virion from getting into the cell. On the host side, host targeting therapies are going to be drugs that will dampen that over hyper-inflammatory response. So dexamethasone is a drug that is used frequently now on this end. This is a corticosteroid, which is a class of molecules that are anti-inflammatory molecules. So based on what we know about the course of the viral infection and the markers of disease, you can envision that when you might want to give a virus targeting therapy could differ from when you might want to give a host targeting therapy. So in the course of disease, early on is when you're going to have most of the viral replication. So either in a symptomatic or asymptomatic infection, this is during the first week or so, when you have peak viral titers, peak viral loads that over time will wane, all right? So patients that are likely going to benefit from virus targeting therapies are going to be patients that are in these early stages of CoV-2 infection and disease, because this is really when the virus is amplifying, all right? But those patients that progress to severe COVID-19, remember at this stage of the disease, well, the virus has already largely been brought under control, and the problem is not the virus anymore. The problem is the inappropriate host inflammatory response that is coming in and causing collateral damage. So this is the time when patients are likely going to benefit from therapies to dampen immune activation. If you give them antiviral therapies at this time, it probably isn't going to do much because the virus is largely gone then. Similarly, if you give immune dampening drugs too early, you can envision that would be dangerous because having a robust immune response early is really important to control infection. You just want to dampen that later on when it becomes immunopathological, essentially. All right, so At this time, unfortunately, none of the drugs that are being used or therapies that are being used in the current studies, at least under the regimens that they've been tested, have been shown to basically be the magic bullet to stop COVID-19. And so what we really need are either new treatment regimens or ideally a vaccine to protect us from getting infection or disease in the first place. So how do you design an effective vaccine? And what's the difference between a vaccine and a natural infection? Essentially, an effective vaccine is something that educates the immune system to recognize essential viral antigens whose inactivation is going to block infection. So let me explain to you what an antigen is. An antigen is any bit of the virus as it's getting chopped up and viewed by the immune system, any particular bit of the virus that is foreign. The problem is your immune system doesn't a priori know which bit is something that if it directs an antibody against that bit is going to stop the virus. So let me make an analogy to a car here, in which if you envision that the virus is the car here, the viral particle, and that is protecting its driver, which is the coronavirus genome. Well, any part of this car could be an antigen that the immune system sees and makes an antibody to. So the immune system may make antibodies to the hood of the car, or the rear tail light of the car, or the front part of the car. Well, these antibodies are not going to stop the car from driving, not going to stop it from moving, even though they're all parts of the car, right? And your immune system doesn't know which part is the one that is really going to be the stopping part. Instead, the type of antibodies that you want your immune system to make are the ones that go to the wheel, right? An antibody there is going to stop the car in its tracks. And that's why we refer to this type of antibody as neutralizing antibodies. These other antibodies are made, but they're not going to neutralize the virus. And in this analogy of the car, the spike protein is the wheel. For the reasons that I explained earlier, its essential role in getting the virus into cells. So there are more than 50 different vaccines for CoV-2 in human trials now. 10 are in phase 3 trials as of this month. I just checked a few days ago. And because I'm a gene expression person, I tend to group these into two general categories. Category 1, shown here on the left, are vaccines in which we are basically producing the spike protein and giving that spike protein to the host, injecting it into an individual to basically tell the immune system, make your antibodies against this. This is what's going to neutralize the virus. And so we can either do that, we as scientists or medical personnel can do that by purifying the spike protein, growing it in the laboratory and just having the spike protein itself that gets injected, or by growing up batches of the virus and inactivating, essentially killing the virus so that the spike protein remains there and can be seen by the immune system, even though the virus can't replicate. The other class of vaccines are ones where instead of giving the immune system the protein itself, what they do is give your body the instructions for how to make the spike protein itself. So they're giving the set of genomic instructions and having your own cells make spike protein to present to the immune system instead of delivering the protein in the vaccine. And so the two classes of vaccines in phase 3 trials here are those that have sort of a safe vector backbone. So several of them are using a safe adenovirus vector. This is a virus that has been made unable to replicate in human cells. And they've put the sequence encoding that spike gene inside the adenovirus. So the adenovirus basically serves as a delivery mechanism to deliver that gene to the nucleus of your cells, where it can then be made into messenger RNA, and your cell's protein synthesis machinery can turn it into spike protein to show to the immune system. The other one are these messenger RNA vaccines that you've surely heard a lot about in the news. Here, the sequence encoding the spike protein itself is just delivered on a snippet of RNA that is then basically entered into the cytoplasm of cells through a liposome and can be directly made into spike protein. For all of these viruses then, this spike protein, once it's presented, is taken up by cells that are part of your innate immune response that are antigen presenting cells like dendritic cells. These cells then show the bits of the spike protein to your adaptive immune system using helper T cells, which will then communicate to your B cells and say, hey, make a lot of antibodies against this, or to cytotoxic T cells, which will be used to recognize already infected cells and to kill them. So we're still waiting for the results from the phase 3 trials. We should hear about them within a matter of weeks to months, so very soon on the horizon. But as we wait for those data to come in, the good news is that there are some things that we can do now, available now as strategies to prevent COVID-19. And those are wear a mask, wash your hands, and keep your distance. So with that, I'm going to stop, and I'm happy to take any questions from the audience. Thank you so much for listening.
Great. Well, thank you so much, Britt. That was awesome. Brilliant, brilliant talk. I'm going to steal a little bit of the host's prerogative and ask a question real quick here before the others start to come in. So you gave us really interesting information about how background genes in people could play a role in why some people have severe responses and why others don't. I'm also curious as to why certain risk factors, age and certain ongoing diseases like diabetes play a role in why some people have severity. And you want to comment on that a little bit?
Yeah, I think that's a really interesting question, and also something that is being extremely intensively studied. Simplistically, how I guess I would think about it is that these, what we would call comorbidities, which are pre-existing conditions basically, or advancing age, are things that can be associated with decreased immune function. So we know that as people age, their immune responses tend to be somewhat less than an individual who is young. And if you have a pre-existing condition, certain types of diseases, also obesity is a situation where we know that people have immune systems that do not function to their full capacity under situations of extreme obesity. So these are things that tie still back into this idea of these conditions prevent you from mounting as effective of an immune response early on as you can. There could well be other things that are sort of fine-tuned interactions with the virus that favor a severe versus a mild infection as well. But I think of it as, sort of, it probably all comes back to how effectively your immune system can respond, particularly early on.
The innate one early.
That's correct.
Got it. All right. Well, let's see. Alyssa, do you want to start providing us with some questions that you've collected there?
Yeah, I'll go ahead and jump in here. We do have a handful of questions, a couple that came up early in your presentation. Can you speak to the level of contagion of this virus? Someone asked, is the rapid replication of the virus the reason that it is extremely contagious?
Probably not so much the rapid replication. Many RNA viruses tend to replicate pretty quickly. The levels of viral RNA that can accumulate in an infected individual, though, are very high, up to 1 trillion copies of RNA per milliliter. Okay, 1 trillion copies when you are at your peak infection. So that means there's a lot of viral RNA there. That's not all infectious virus, but that does mean that, especially because the virus is replicating, when it is replicating in your respiratory tract, well, you're going to breathe it out. And so when you're in your peak infectious period, there's a lot of potential for transmission that way. And so viruses that are transmitted through respiratory droplets often can be more easily transmitted than other types of viruses. And so I think the transmission for coronavirus is efficient in part because it's a respiratory virus, perhaps in part because the cells that it can infect may be cells that it immediately encounters once somebody sort of sucks it in or breathes it in that way.
Thank you. Okay, so our next question that comes in asks, is it possible to reduce the overcorrected immune response without causing further damage to the body?
Yeah, one would hope, and this is the idea behind these host-directed anti-inflammatory basic treatments, so things like dexamethasone. The idea is, can you reduce that inflammatory response when it isn't helping you anymore? In fact, it's causing tissue damage. And so that's where I think anti-inflammatories can play a role when given to individuals who are sort of progressed towards this more severe phase of the disease. Other ideas that are being explored are potentially being able to boost that early immune response, right? Instead of dampening the immune response late, well, if severe disease is correlated with a failure to mount a robust early immune response, might you be able to treat individuals with things like interferon very early on and help them then have sort of a resolved infection that is not associated with severe disease?
Thank you. Okay, so there are a few questions asking about how the coronavirus compares to the common cold. Charlie Arnson asks, he's seen a couple of publications that provide evidence that prior exposure to the cold virus, coronaviruses can give partial protection against SARS-CoV-2, which might explain why, it's important for kids who have more common colds. Is this commonly accepted?
Yeah, so this is a really interesting question. He's correct that there have been a few studies now that have come out that have looked to see whether or not there are components of our immune system, be they antibodies, that you would have gotten perhaps because of prior exposure to one of the circulating coronaviruses that are associated with the common cold that are related to COVID-2, but don't cause that same severe disease. Or T cells, whether there are T cell epitopes or antigens, I guess, that are recognized by T cells that can cross-react with components of the common cold coronaviruses and COVID-2. And so these studies have suggested that indeed there are some cases where individuals will have parts of their adaptive immune system that have been exposed to common cold coronaviruses and may have cross-reactivity to COVID-2. The important distinction, though, is that while those exist, it is not yet known whether that cross-reactivity actually confers protection against COVID-2. So that is a hypothesis, and it's a really interesting hypothesis and one that people are designing studies now to test. But we don't know whether that protection, because those, for example, in the antibody examples that I gave, well, those antibodies might not bind the COVID-2 spike protein in a way that neutralizes it, even if they bound the circulating coronavirus spike protein in a way that might have been neutralizing. So that's a really important question and area that is being explored now.
Awesome. We might have time for one or two more. I see a couple of questions that bring up herd immunity, if this is possible, and kind of asking what are the future implications of this pandemic if a vaccine is not available in the next few years. Can you speak to that a little bit?
Yeah, I think the herd immunity question is an extremely important one because almost all medical personnel and virologists are on the same page on this one, which is that it is a very bad idea. And the reason that it is a very bad idea, well, there are multiple reasons. One of the reasons is that it's estimated that you have to get, 50 to 60% or more of the population infected in order to potentially confer herd immunity. We are far, far below that level right now, and we're already at 200,000 deaths in the US. So if we tried to quintuple our infection rate to achieve herd immunity, we'd be reaching 1 million or more deaths, and that is not worth it. That is not a viable strategy to protect the population. The other problem is that, we don't yet have evidence of, there's almost no situation of natural infection leading to effective herd immunity. the examples that we have where herd immunity works is because of vaccine-mediated protection. So measles, for example, or there are other examples where it has worked, but it has been through the delivery of vaccines. So only a vaccine-mediated strategy to generate herd immunity will be safe. If we try and do it through natural infection, there is going to be a huge amount of death in this country and around the world. So I would say that is why herd immunity is a bad idea through natural infection. Trying to achieve it through vaccination, though, I think is a great idea, and that's something that we're hoping to do. The second part of the question, which is what might the landscape of this pandemic look like, depending on the outcome of these phase 3 clinical trials for the vaccine. I think, most of us are very, cautiously optimistic that the vaccine data are going to come back and look promising, but that doesn't necessarily, we don't know that that's going to be true. I mean, the thing about science is you can hope anything that you want to hope, and the data are going to tell you whether it works or not. And this is really important to think about. This is a virus that has been highly politicized, and whether people want to wear masks or take the vaccine or trust the government or things like that, and that's all very understandable in this current political climate. But I think it's important to understand that the science it doesn't take a side. It doesn't matter whether you're Republican or Democrat or Libertarian or whatever. The data are going to look like what they're going to look like, and those data are going to be evaluated by people who have the expertise to look at knowing exactly what are the markers that one has to evaluate to determine if the vaccine is efficacious and if it's safe. And so I will, and all the rest of the scientific world will be looking to that analysis of the data, which are going to either give us a yes or a no. And if they tell us that they're safe, the data say that the vaccine is efficacious and safe, well, I'm going to be lining up to take it. But we may not know that right away. And there is, of course, a chance that these data will tell us that this first round of vaccines aren't going to work. In that case, then we may have a longer wait before we can get a vaccine. And in that case, I think, we're really going to have to rely on, first and foremost, the tried and true public health measures that have been used to keep people as safe as possible in pandemics throughout human history, which is, the masks and the hygiene and the distancing. And so trying to maintain that level of safety as best as possible while continuing to do research to search for new antivirals or new ways of delivering existing antivirals so that we can get better and better tools for fighting the disease for individuals who get it, even if we don't yet have a vaccine that can prevent us from getting infected in the first place.
I'm going to hop in with one last quick question. Do you want to comment a little bit about where this thing came from?
Yeah, it's pretty clear from there are hundreds of thousands now of sequences that have been deposited of the more than any other virus in the data banks. And so people who do evolutionary analysis can trace where these came from to some extent. It's pretty clear that it originated in bats. What isn't clear and may never become clear, I guess, is that whether it came directly from bats to humans. Bats tend to be reservoirs for many, many different human pathogens for reasons that are quite interesting and we don't have enough hours to go into right now. Or frequently, viruses will jump from bats into what's called an intermediate host or hosts, where they will then acquire some more changes as they're replicating in that intermediate host to jump to a human. And so there's been a lot of discussion that pangolin could be that intermediate host for CoV-2 because the most closely related coronavirus sequences are to pangolin coronavirus sequences. So one hypothesis is that it went from bats to pangolins to humans. All that's really certain is that at some point it came from a bat, and what were the intermediate steps, we don't yet know.
Great.
It was definitely not engineered in a lab, though. That is eminently clear from the data.
Exactly. Thank you. Well, listen, we can't thank you enough, Britt. That was just a phenomenal talk and wonderful questions and answers. Want to thank everybody who came tonight and who listened to this phenomenal science. If you enjoyed this event, I want to remind you that the Biodesign sponsors SIPA science events where well, in the past we've been going to restaurants and do presentations. I think this year we'll be doing them virtually, but we have a variety of researchers lined up for the winter series, where we give all kinds of talks about all kinds of really interesting topics. We want to encourage anyone in Arizona to go get tested on a regular basis if that will help you. Certainly, if you have a public-facing job and you interact with different people every day, it's a good idea to get tested on a regular basis. They're free tests, they're saliva tests, so there's no sticks in your nose. This talk will be available on our website. And of course, our website also sponsors statistics about what's going on with current infection in Arizona and about Arizona. We welcome anyone who's interested in the biodesign to join our Pioneer Circle. Visit our website to track the pandemic in our state. And please stay safe. We have the holidays coming. This is not the year to bring the entire extended family together. Maybe keep the family meetings a little bit smaller this year. Visit people by Zoom or by phone or whatever you have to do, and follow all the mitigation factors that Britt so carefully pointed out to you. So stay safe, everyone. Stay healthy. It's a pleasure to have you all. Take care.